Alright, so this is our PowerPoint on homeostasis and feedback loops. And the main question we're going to ask today, how do our bodies or our cells know when our internal conditions are changing? Okay, we know that homeostasis is one of the characteristics that all living things have. The definition is that it's the ability to maintain relatively stable internal conditions. Okay, so the way that the cell is able to control these conditions. One example would be body temperature. Okay, think about what happens when it gets too warm. Think about what happens when it gets too cold. We've already talked about this before. You would sweat or you would shiver. But the question is, when does your body know when to switch? When does your body know, have you sweated enough and has your temperature returned to normal? Okay. It's going to use a feedback loop. A feedback loop is a signal or a mechanism that's going to help start or stop a process. Think about a thermostat in your house. Okay, how does it know when it's going to heat the air or not? Well, the thermostat contains a sensor, and that's going to tell when the temperature in the house has changed. Okay, so if the temperature starts going too low in the house, then you're going to hear click and your heating system will turn on. But it's not going to stay on indefinitely. It will stay on until it reaches that, before temperature, and then it will shut off once it reaches that desired temp. Okay, so here's an example of a feedback loop. We have four main pieces. First is the environment. It's going to produce a stimulus, and a stimulus is just something that causes the need for a reaction. For example, if you get stung by a bee, the stimulus is that bee sting, and yes, you are going to react to it. Okay, so the environment produces some sort of stimulus, in other words, a change. Okay. The receptor is within your body, within different parts of your cells, and that is what's going to respond to the stimulus. In other words, it receives the signal. Okay, the control center could be an organ, it could be a part of the cell or a certain type of cell, it doesn't matter. But the control center is what receives that signal from the receptor, and it determines what to do about it. So it picks what the response should be. So it's like the deciding factor. And then last is the effector, and the effector is the one that carries out whatever response the control center has chosen. Okay, a lot of times we would see this in your muscles or in your glandular cells. So those are the four main parts of a feedback loop. Different feedback loops have, different pieces in them. You could draw a feedback loop with 3 parts. You could draw it with 6 parts. We feel as though 4 parts sort of explains just about every possibility that we're interested in, specifically within the human body. Okay, so let's go back to our thermostat. All right, the temperature of the room is going to be our environment. If that changes, then it's that stimulus. It's going to cause something to be adjusted. So then there's going to be a receptor, right? The fact that our temperature has changed needs to be received by the sensor, which is in the thermostat. Okay, once the sensor has been, has been triggered, so to speak, then the control center, that is sort of the brains of the machinery, the control center decides whether we need to turn the heat on or off. Okay, it tells, the heat source, which is the effector, it tells the heat source to turn on, the heat is produced as part of the response, and the temperature of the room rises. So as you can see, it's sort of this chain of events that is going to bring our conditions back to their stable environment. Okay. This is known as a negative feedback loop. So the goal, just like a thermostat, is that we are trying to prevent any changes in the environment. We want it to be stable, return to normal. Think of that. Okay, so we talked about the thermostat already. Let's look at insulin regulation as an example in the human body. All right, in maintaining homeostasis, we need to control the amount of sugar that is in your blood. And hormones, which we know are proteins that are used for signaling between cells, hormones are going to play a huge role in the regulation of insulin. In order to control the amount of glucose in your bloodstream, your blood sugar, the body uses a negative feedback loop. So we're going to go through the stages of this with a diagram. All right, so first, the stimulus. Okay, over here. Let's say you just finished a nice, fatty, sugary meal. Okay, you went to McDonald's and you got, a Big Mac with cheese and French fries and a soda. Okay, as all of that food is being digested in your stomach, it's going to be released down into your small intestine, and from your small intestine, it's going to diffuse or move out of the small intestine into your bloodstream. Now, it's been broken down into its individual monomers. So you have individual protein monomers like amino acids, and you also have glucose monomers as well. As the blood glucose rises, okay, there's going to be a need to respond to that because we want to maintain a stable blood sugar environment. So you've eaten something and there's a lot of glucose in the bloodstream. That's your stimulus. Receptor. There are cells that are going to receive this signal and they're going to send a message to the pancreas that the level has become too high. There's too much glucose in the bloodstream. So the pancreatic cells, they're specifically beta cells if you're interested, they are going to act as the control center because they will decide what is going to happen. At the control center, they decide to release the hormone insulin. Okay, so little tiny signaling protein molecules. And what effect does insulin have? Well, it causes your body cells over here to take in glucose. So it's going to remove excess glucose from the bloodstream. If there's still too much glucose, then it will store it as well in the liver. Okay? So your body cells and your liver are going to help remove glucose from the bloodstream. As that happens, then the blood sugar levels return to normal and the process stops. So we don't need to keep circling round and round. We just wait until there's another change in our blood sugar level. Okay, now let's look at what happens if the blood sugar is too low. Okay, here the stimulus is that our glucose levels have fallen. So this would be, for example, if you are, if you're looking into, you haven't eaten in, say over 3 or 4 hours. Okay, all of that glucose has been broken down by your cells, and there's a very low level in the bloodstream. Receptors, again, we're going to have cells that are going to send out alert signals to the pancreas to say, hey, our blood sugar is too low. Again, in the control center, it will be the pancreas, but this time, we're not going to release insulin. We're going to release a different hormone called glucagon, and it's going to be released from the pancreas. And the glucagon is going to travel directly to the liver. Okay, and once it gets to the liver, it's going to encourage the liver to release all of that stored glucose. Remember, when we store glucose as animal cells, we store it in the form of glycogen. So that glucagon hormone will help break the glycogen down into glucose and allow it to enter back into the bloodstream, thus causing a rise in your blood sugar levels so that they return back to normal. And then the process is going to stop. So it's negative feedback. We are keeping control to return to a stable environment. Okay. There are examples of positive feedback loops. These are much less frequent and they sort of have the opposite effect. The idea is an environmental stimulus has been received and the goal is to strengthen that stimulus, 10 times, 100 times, 1,000 times. You want to increase that change in the environment. Okay, so instead here, we're going to accelerate our stimulus rather than try and stabilize and return that level back to normal. 2 examples, blood clotting, which we're going to look at in a second, and labor contractions. Okay, before we go into the examples, I want to give you sort of something a little more applicable to make you understand positive feedback. For example, think about poison ivy, okay? Because of the poison, your skin itches. What do you want to do? You want to itch it. And so you itch it, okay? But by itching it, what that's doing is that is releasing a lot of hormones and a lot of signaling proteins that is going to trigger further itching need, okay? So you're itching, and then the purpose is that it makes you itch more. It's not that by itching you go, oh, wow, that's a relief, okay? This is positive feedback. Instead, as you itch, it's going to increase that signal being sent to your nerve cells, and that's going to cause you to itch even more. Okay, so keep that in mind. We're trying to enhance or accelerate the signal we're receiving from the environment. Down here is a little diagram showing you what happens during labor. Okay, here we see the fetus. What happens is we receive a physical signal from the environment. The head starts to push against the wall of the uterus down at the bottom here. Okay, from that physical pressure, there are going to be hormones released, specifically known as oxytocin. And what that's going to do is that's going to send a signal to a gland to release that oxytocin. It's going to carry it through the bloodstream, but the releasing of that oxytocin causes contractions to occur, okay? And the contractions are, the squeezing of the uterus lining, okay? And what does that do? That pushes that head even further down onto the uterus, the opening of the uterus. And so as you can see, the head pushing releases a hormone which increases the amount of head pushing. The idea is we want to amplify or accelerate that signal until ultimately the baby is born. Okay, let's look at another example here. Here's a blood clotting diagram. Okay, step one, there's been some tear or some break that has occurred in a blood vessel wall. So that's a stimulus. Okay, from this, cells release chemical signals. Specifically, these cells are known as platelets. Okay, and platelets are little fragments of cells that are responsible for clotting your blood. So they're going to come to sort of the scene of the crime, if you will, and they're going to stick. Okay, once they stick to the area, they're going to release more chemicals or more signals, which are going to tell other platelets to come and join them. So we're increasing or we're amplifying the signal as the clotting is going to occur. We get platelets that arrive, okay, and once they stick to start patching this up, they release chemicals calling for more platelets to arrive to stick to help patch things up. And this is going to circle over and over again, getting stronger and stronger to increase the number of platelets that are clotting. That is until the clot has been healed in some way. Okay, so we've looked at two different types of feedback systems, and we see how important the maintenance of life is, or how important it is to maintain certain chemicals and things like that in our body. Lots of things are under control from feedback loops. For example, heart rate, okay, your blood pressure, breathing, the amount of oxygen and carbon dioxide being measured in your bloodstream, and other things such as minerals and ions, calcium, things like that as well. So maintaining stable conditions for the most part, okay, maintaining meaning the negative feedback loops, those are going to be very important. So how on earth does the body, control this? How can we look at this on a cellular level? Can you think of any cell organelle that we have talked about that is going to play a role in controlling or regulating movement of things such as proteins or signals? Hopefully you came to the cell membrane, okay? We're going to see that the cell membrane plays a very important role in homeostasis. It is going to help us maintain balance of things like water, other solutes like salts, amino acids, glucose, things like that in and out of the cells. And generally when we are looking for homeostasis, we wind up with this situation here on the very end where we want to keep a balance of particles inside and outside of the cell. But how does it know when these particles should be moving? We need those signaling proteins, those hormones. Okay? So this is sort of a lead into our next section, which is going to talk about cell transport. Take a second here. There's 2 links on the bottom. See if you can get them. They are Shockwave flash players. So if you do not have Adobe Shockwave, you're going to need to download that in order to play it. If you can't get it on your computer at home, feel free to come in before school or after school, and I can let you borrow the laptop so that you can view them. The first animation is a simplistic step-by-step through the blood glucose homeostasis. The second one goes into a little bit more depth and explanation. So make sure that you are able to describe the pieces of a feedback loop, because in class what we're going to be doing is we're going to be looking at other examples of homeostasis in the body, and you're going to have to try and figure out where those 4 pieces fit. We have the stimulus, the receptor, the control center, and the effector. Okay? All right, thank you very much.